Thanks.
Hello, everyone. Thank you for joining this seminar series organized by the Empathic Computing Laboratory. I'm your host, Aladdin, and our distinguished speaker for today is Laura Angeletti. I hope I said that correctly, who is currently a licensed psychologist, PhD candidate, and a research fellow at the International Research Center for Cognitive Applied Neuroscience. Laura has did a lot of work on behavioral neuroscience, empathy, and neurophysiological measure, which all of these we are interested here in the lab. The title of the talk is an hyperscanning paradigm to investigate social interoception. The talk will be roughly an hour, and I think Laura, she has to leave after an hour. But thank you for sharing your time with us. Thank you.
Thank you, Aladdin. Nice to meet you all and to be here. I will, thanks for the introduction. I will try to share my slides for today, my presentation. Okay. I think now you should all see the full screen presentation, right? Okay, so good afternoon to everyone. I know that for you is afternoon, while for me here in Italy is just 8 a.m. I'm very happy to be here with you for this talk today. And thanks so much for inviting me to this seminar series. My name is Laura Angeletti. I'm a PhD candidate and a research fellow at the International Research Center for Cognitive Applied Neuroscience. And actually, I work here at the Department of Psychology. I'm a psychologist, I'm not an engineer. So I basically conduct research in neuroscience from this perspective. Today, I'm here representing my chief, that is Michela Balconi, a professor in cognitive neuroscience. And I'm here also representing the whole group. We all work on different topics. My topic at the moment is dealing with interoception. And the title of this talk, as Dr. Nassani just said, is about an hyperscanning approach to investigate this construct. We will see later why the term social interoception is put in quotation marks at the moment. Just to start, and also I want to say that for any curiosity or question about what we do at our center, here I leave the website, so it's psychoneuronet.com. Just to start with a brief introduction, first of all, let me introduce where the center and the laboratory is and are based here in Italy. We are here where there is the red dot, and it's in the northern part of Italy. Our lab is inside the university, is inside the Catholic University of the Sacred Earth in Milan. That is this beautiful building. Of course, we have also, as lots of Italian university has beautiful clusters and traditional and classical building and room around us. But actually, we also have a whole building devoted to psychology. And this is the building, the one on the top, on the bottom right. And in this building, there is a museum dedicated to psychology, and also lots of laboratories for behavioral neuroscience and applied cognitive neuroscience. I don't deal that much with virtual reality, but I know you do. So if you want to take a 4 minutes 360 virtual tour of Milan, here you have a link to do that if you want. As I was saying, I'm here representing Professor Balconi, and at her center, she follows various research lines. She started her career with studying emotions and empathy, but also communication processes, action and interactions dynamics. She also studied basics of consciousness and agency. But in the last and more recent years, she dealt also with some applied research fields, such as you can see here, neurocognitive empowerment, but also neurolaw, neuromanagement. So discipline that relate with management, marketing, and let's say less basic topic. To do so, we aim to explore these different topics. Of course, there are lots of collaborators that and different collaborators from me that treat these topics. We use several different neuroscientific tools, as you can see on the right. Indeed, at the lab, these are only some of the tools that we use and we have. We have different models of EEG, from more fixed to wireless devices. We have also different models of autonomic indices recording devices. We have neurofeedback, fixed and wireless. We also have the wireless and fixed eye tracker. But last but not least, we have functional near-infrared spectroscopy that I don't know if you all know, but it's this tool on the left. So we also have tools for neuromodulation and neurostimulation. So even if our tradition is more classical and more related to the study of clinical psychology and the psychology of family, that's our tradition, we all move to this branch of neuroscience that is about the application to basic and applied research of this neuroscientific tool even outside the laboratory. That's our starting point. As you saw from all the methods that I've just presented, what characterized our approach is a type of specific approach that can be defined multi-method approach to social neuroscience. Indeed, for studying emotion and empathy, that I know is something that is more interesting for you, we always adopt complex research protocols, and these research protocols usually combine multiple measures such as bio, EEG, and FNIRS co-registration paradigm. The reason why we use this approach is that social and affective phenomena are complex phenomena, and they are characterized by reactions that are connected to central but also peripheral nervous system reactions. Therefore, we believe that more tools and more indices integrated together could help us to better explain these phenomena and to study them more deeply. Now, from let's say 2015 on, we developed the new approaches to study the affective and emotional phenomena, and in particular to study social processes. So we moved to hyperscanning paradigms, but we strongly believe that we need new experimental paradigms and even methods, both of research and analysis, to explore first of all, interpersonal relations and interaction that are the focus of our lab, but also to deepen brain-to-brain coupling. So today I propose you to sit together and to speak about the application of a relatively new tool that we use in hyperscanning, that is FNIRS. It is, let's say, relatively new because we just started to go in depth with these, with more complex analysis and more complex paradigm. But we have this tool from at least, as you can see, 6 or 7 years. The professor used it a lot in her studies, and what we will do, it will be to see 3 examples of research protocols that apply this tool while collecting data in a hyperscanning paradigm. I don't know if you all know what is functional near-infrared spectroscopy, so I will just tell you this briefly because I think most of you are bioengineering researcher and professor. This tool is an optical imaging technique, so it uses light to explore and investigate the cerebral blood flow. It is in this case used as in the field of neuroscience as a neuroimaging tool, and in general it allows to investigate the activity of different cortical areas, in particular their hemodynamic responses during a task or when facing a specific stimulus or a specific condition. What it gives us is a map of the neurofunctional activation related to the mainly to the cerebral cortex and not to related to subcortical structures. Why to use this tool in hyperscanning? We now know that it is more easy to use for cognitive, emotional, and in particular for motor tasks during complex interactions, because it's in particular less sensitive to motor artifacts. So for us is easy to use compared to fMRI. It has low physical and experimental limitation, and it preserves a high ecological validity to study interactive and dynamic social phenomena. And of course, it's less invasive. It has been used for different cases as an alternative to fMRI, or also from PET for to avoid radiation on liquid injection. And it has been used also for fragile samples in the clinical field. So it is used for newborns, psychiatric patients, or elderly. There are so several advantages to use this tool. We usually split the channel that and the optodes and the detectors and sources we have onto different brains to collect the brain activity of the two members of the dyads that interact in a condition. As I said, it's not a new tool for us, even if there are not so many studies exploring hyperscanning paradigms using NIRS. Compared to other techniques, like compared to EEG hyperscanning studies. In the first article here on the top right, the professor and her colleague explored the past, the present, and the future of the application of NIRS to study emotion and social neuroscience. And NIRS have been so far applied to emotions, in particular to emotion recognition and emotion appraisal, but also, as I said before, to clinical population with emotional disorders. It has been lately been applied to social neuroscience and to social context, like interpersonal dynamics, but also in particular communication or nonverbal processes and joint actions. And in this article in 2016, authors said that some future application of NIRS would have been the application of this tool to study brain plasticity and hyperscanning and hypercognitivity. That's where we arrived now. More recently, indeed, we have published this article that is about exploring the connected brain by NIRS, by studying human-to-human interaction. And in this article, we have a collection of the different study and evidence in which we applied NIRS in hyperscanning. And we also deepened the single brain and dual brain dynamics and the different type of analysis that we applied. Here I've just reported some examples of how nowadays we use the NIRS in our lab. Indeed, starting from the top left, hyperscanning paradigms adopting NIRS were really used for investigate mother and child relations. In general, they have been used to investigate relations, cooperative dynamic, but also social gains and creativity during young peers relations. In our lab, it has been used for studying in general cooperative and competitive dynamic. And in some cases, we moved from the study of basic cooperation and competitive dynamic outside the lab, studying these dynamics in other applied contexts, such as sports or teamwork, also managerial teamwork. Also, I have to say that some colleagues of mine use FNIRS hyperscanning paradigms to study more prosocial behavior and empathy, such as, for example, my colleague just studied the difference and published some articles about the difference in receiving and donating a gift, so a dynamical gift exchange, to investigate the different cerebral area that process gift exchange and donation. But today we will not focus on this specific topic about gift exchange. As I said before, we will see instead 3 examples of NIRS hyperscanning application to 3 different topics, to explore 3 different social dynamics. First, we will describe this study about cooperation and competition. We will then see briefly the effect of interbrain synchronization in gesture observation and reproduction. So there is also this study about my colleague and Professor Balconi related to joint gestures. And at the end, as I anticipated, we will speak about social interoception. We don't have an article about this topic because it's an ongoing study. So it's the first time we present this work to somebody. And so the best is always at the end. And about the joint gestures and cooperation and competition studies are premises necessary. Indeed, I've not dealt directly with these 2 studies, but the professor really asked me to present something else except the study I'm performing right now. So I will give you some general indication and lines of other projects that we conduct, and we also my colleagues conducted before me. Okay, so let's start from the first study. This study, as I said, is all published already in this PLOS ONE article. It is about the effects of strategic cooperation on intra and interbrain cognitivity by FNIRS, and it has been conducted with some colleagues from France. Here the authors studied interpersonal brain-to-brain coupling during a specific social task requiring both either cooperation or competition. But we will see now how they manipulate this condition. We will start briefly from the theoretical background, just to give you some path to follow. And as I said, I will not go so in depth with these topics because they are not my field. But I want to say that the first question was for this author was which dispositional mode is better, let's say, for the brain between cooperation and competition. So they wanted in general to deepen the effects of cooperation, either competition, on different levels of complexity. That's the reason why they applied questionnaires, hyperscanning paradigm with bio, EEG, and FNIRS co-registration to investigate the self-perception of a person while it's cooperating, either competing. They wanted to serve with some manipulation check the self and other representation during the social interaction. But thanks to a specific task, joint task, of course, they wanted to explore even the cognitive performance by behavioral measures, so reaction time and rates. The brain correlates of the participant while performing this task. And at the end, they also perform interbrain cognitivity analysis. The reason for these analyses were possible because, as I said, we were performing an hyperscanning paradigm. In some cases, there are different answers to this question are collected in different articles, some about cooperation and others related to competition. But today I will describe the whole methodological implant and the task as if it is composed. There are two different tasks, but I will try to explain everything together to make clear how what changed between one condition and the other condition, because the task was shared between these two studies, and it will become more clearer later. As we said, I don't know if you goes in depth before with cooperation and competition dynamics, but these are two basic modes of interpersonal interaction. In a sense, they both require an empathic point of view and self-monitoring abilities, because the person needs to reciprocally manage the other person's actions in terms to adapt one's own. In some cases, of course, they have to cooperate, so they have to combine their behavior and their choices. But also in case of competition, they have the participants need to have a well-developed theory of mind to understand how to perform better than the other person. In this sense, they activate different aspects of empathic processes, and they call on different social processes during the mentalizing processing of the situational behavior they are performing. The thing is that the agent need to determine the other agent's mental state to understand and to predict what will be better to do in the next action or in the next choice or behavioral act. We can just start saying that in general, in competition, while competing, a person needs to put higher mental effort because the goals of the person becomes of the dyad become divergent, and so we saw, and also other colleagues saw, that competition require a general higher activation in the brain. As I was saying, there are previous authors like DCT and colleagues that studied the effect of competition on the brain. And what they saw was that in general, both competition and cooperation activates a bilateral, they saw a bilateral activation of the anterior insula when participants cooperate and compete. But as I was saying more in general, now we can see that in this picture, when a person is competing, in this case, the CT found a medial prefrontal cortex activation that was related to a greater mental effort and a greater activation of TOM abilities. That's something that we also found in the studies on competition, but what we let's say conclude was that this mechanism, both these dispositional modes, rely on executive functions, and specifically the person, the two members of the dyads deliberately select a salient knowledge or a salient response to achieve an internal goal. In the case of competition, the difference is that the opponent's behavior is less predictable than in a cooperative dynamic, and because there is not a planned expectation of the other agent's behavior. So we observed an increase in prefrontal cortex activity, and in particular of dorsolateral prefrontal cortex activity. While we know that in general, when dyad cooperate, the behavior and the brain activation is facilitated because the behavior of the other agent is clear and at the disposal of the participant of the other agent. Given these premises about competition, we will now then focus more on some results related to cooperation. Indeed, the study that the results I will briefly discuss will be related to brain-to-brain coupling during cooperation. To do so, the authors implemented an hyperscanning experiment, as I said, and this is the task they used. The authors presented these participants a task, a simple experimental task, in which they were required to select a target stimulus between some non-targets. And these targets were based on 4 different options of shape and color. And the task is relatively simple. They see the target at the beginning of a trial of a trial of 25 trials, and of a block of 25 trials, and they know they have to press the space bar when they see the target, and to avoid to press the space bar when another target is shown. After 3 trials, they received a trial feedback. And after this block, they need to fill in some quick question about their self-perception. At the end of 50 trials, there is a halfway feedback. So as you see, the task is relatively simple. What is interesting is that the authors use this task in 2 different conditions by manipulating the instruction that they gave to the participants. So they, in one case, participants were asked to cooperate at the beginning of the task. So they received this instruction: try to cooperate and synchronize your response. And this will be a measure to evaluate your subjective skills of teamwork. And after each trial, let's say if each block of trials, they receive an evaluation, a social feedback related to their cooperative score. And it could be 2 arrows up that was related to high cooperation score, minus related to a mean performance, or 2 arrows down that is a low cooperation score. This was the manipulation for the cooperation condition, let's say for the cooperation study, because these 2 tasks were divided. After that, the professor and colleagues chose to focus on competition, and they wanted also to manipulate in a different way this instruction, and they asked the participant to try to perform better as they can and more than their competitor, and this would have been a measure of proficiency of their skills. And also in this case, they received a social feedback of their performance with 2 arrows up for a better performance, a minus for a comparable performance between the two agents with the competitor, and 2 arrows down as a worse performance. And of course, this was evaluated on the basis of the performance of both members of the dyad. This was a multiple measure study to, in general, as I said at the beginning, to investigate the brain correlates also from the electrophysiological perspective, not only from the neurophysiological and neuroimaging one. We also applied some self-reports like locus of control behavior, behavioral activation system scale, but in this case, we will not see all these results. We will focus on the FNIRS evidence. What we saw in general terms was that in this case, main FNIRS evidence for cooperation was that during cooperative behavior, there was an increase of left dorsal prefrontal cortex activity, and also to check all the details about where channel was positioned to map this area. You can find all the description on the article. What we found was that this left activation was detected both in pre and post feedback, but was higher in the post feedback condition. This was an important effect for the authors because this is a lateralization effect related to the left portion of the prefrontal cortex. And it was interesting because we know, according to the Davidson theories, that left prefrontal cortex in general is more related to the processing of positive emotions and approach attitude. And in this case, this result was interpreted in this direction, like as if during a cooperative dynamic, this increased in left dorsolateral prefrontal cortex signaled a positive emotional processing of the dynamic and an approach attitude versus towards the other partner. The most interesting thing is that compared to the right hemisphere, there was a presence of a left hemispherical effect. So this lateralization effect was detected both in EEG and FNIRS measure and was associated to the cooperative dynamic. Once again, after the post-social feedback, this effect was higher. This is the correlation between the couples that were involved in these studies, and this data refer to the oxygenated hemoglobin mainly. They didn't find particular effects for the oxygenated hemoglobin, and in general, what we can say that is that for this, let's say, set of studies, more interesting result was detected for the oxygenated signal than the deoxygenated one. So these in general are the main results found for these first studies. I know they are general lines, but what's interesting to point is this lateralized hemispherical effect that is unbalanced towards the left sides. And of course, these are just basic studies. It will be interesting to check the same dispositional modes with more ecological tasks. That will be one of our aim. We know that there are other interesting cooperative tasks, but we do believe that in some cases also the instructions that we gave and that are given at the beginning of the task can really deeply influence the real dispositional modes in which the participants face the tasks. So as you can see, this task could have been performed both competing and cooperating, but what's the main difference? The main difference was the instruction given at the beginning. Okay, this was the first studies, and I can answer to some questions later if there are. I hope there are some questions. There will be some questions, but I can leave this part at the end if you agree. So we can go on with the recording.
Yep. If anyone has a question, please feel free to ask. Otherwise, we can wait until the end as well. I had a question about the last graph. So there was a different graph between cooperation and competition, or is it the same O2 HP here? Was there a difference?
Do you mean here?
Mm.
Okay, this is on the left, it's only the result of cooperation, and on the right is just an example, a figure. Is not the evident cluster that we use. It's just an example to show you a similar activation, but we do have only this graph.
Okay.
On the left.
Okay.
Okay, I can just show you the second study that is about joint gestures, and also this second study is relating to an interesting social process that is in this case the human ability to develop a strategic and joint pattern of non-verbal communication signals when we communicate. In this case, we use once again a NIRS IP scanning paradigm, but to investigate the neural responses and the interbrain synchronization associated to gesture communication. In this case, the task was divided in two subtasks. There were a first task related to a gesture passive observation of the different non-verbal signal, and another subtask related to gesture reproduction. Specifically, in this case, the Professor Balconi and my colleague aim to investigate the both, as I said, the neural response and interbrain synchronization processes associated to gestural communication. But in particular to this type of communication during three specific categories of gestures. These three different categories of gestures were affective, social, and informative gestures with a positive and negative emotional valence. Specifically, there were some video clips that the participants were required to observe in the first case. These videos were related to, for example, in the case of a social positive category of gesture, they were gesture related to start or maintaining a relationship with the other agent. Or on the contrary, in the case of a social negative gesture, the gesture was used to stop the interaction, and also, as an example, really the stop gesture was one of the gestures included in this category. Instead, the videos reproducing positive and negative affective gesture were more related to the interest of communicating physiological, psychological, or physical state of well-being of the interlocutor. So, for example, the gesture of saying that he felt pain in some parts of the body was a negative affective gesture. And finally, there were some informative gestures whose valence were defined by the description that appeared at the beginning by a context slide presented before the video. So they were just informative, but manipulated in a positive or negative valence, depending from the context. The participants were, as I said, involved in this hyperscanning paradigm, and they could have two roles during the gesture reproduction tasks. In one case, there was an encoder who was asked to observe and reproduce the gesture observed in the video and to reproduce this gesture towards his or her partner. And there was a decoder who was instead just asked to observe and receive the gesture that was reproduced by the encoder. So in the gesture reproduction subtask, there was this different dynamic. And what we observe, even in this case, we collected the data related to the neural response, both oxygenating and deoxygenated hemoglobin, by using the hyperscanning paradigm. In this case, my colleagues and Professor Balconi, and here I can just report some general effects divided to the subtasks, observed, for instance, in the top left graph, that there were different portions of the cortex that differently responds to different category of gesture reproduction. So, for instance, there was a higher presence of oxygenated hemoglobin activation in both the participant during the presentation and reproduction of affective gesture in the dorsolateral prefrontal cortex, while it seems that the superior frontal gyrus responds more to social gestures, and a significant activation of the frontal eye fields were found for informative gesture. Once again, there was a left lateralized effect for two specific areas. For the dorsolateral prefrontal cortex, the left portion was more active for positive gestures in general, not divided for any category. And we found a main effect also for the positive gestures in the left superior frontal gyrus compared to the negative gestures. In the gesture observation subtask, instead, here we have a different but still relevant significant effect. Instead, an increased O2HB activity was found in the dorsolateral prefrontal cortex for the affective gestures, and once again, the superior frontal gyrus was significantly active for the social informative gestures. And to confirm the role of the left hemisphere, we found even in the observation of in the gesture observation subtask, an activation of the left dorsolateral prefrontal cortex compared to the right prefrontal cortex, well, not compare, but we found the effect in the left dorsolateral prefrontal cortex that was more respondent and more active compared when the person were observing affective positive gestures compared to other type of the other categories of positive gestures, so social and informative gestures. In this case, we can once again consider the relevance of the left hemisphere in processing mainly social and affective gestures. So these portions are highly responding to affective and social dynamics, and it's the left portion that is responding for that. We can consider these results important in terms also of localization. And what we saw was also that the hyperscanning, FNIRS hyperscanning paradigm was doable and usable even during this simple motor performance. What also we saw is that there is a difference between this type of category of gestures, and these differences in terms of gesture impacts in brain responses, in particular in these prefrontal sub portions. This is once again just an example on how we use this FNIRS hyperscanning approach. Now I will move to the study, to the methodological implant that we just developed to study social interoception. I don't know if you all know the concept of interoception, this type of construct, but I knew for Dr. Nassani that maybe somebody of us could be interested on this. I really hope that after this brief description, you will become even more interested, that because this was for me. I didn't know, before my PhD, I didn't know so well this construct, but now I strongly believe that is really important, in particular also for higher order cognitive processing, even if it's considered a basic physiological type of perception. Indeed, as I said, for those who don't know what is interoception, interoception is a specific type of perception. It has been described and defined as the perception of inner body signals. So, for example, temperature, pain, thirst. And this type of perception, this construct has been recently expanded to in multiple dimensions, as you can see here on the in the right figure, from more basic levels related to afferent signals from the body, to middle indices more related to the behavior of a person that could be more or less able, for instance, to perceive the heartbeats. And these abilities usually measured by heartbeat detection task. And I don't know if you have ever tried to do an heartbeat detection task, but it's really difficult because the person is required to sit and try to count his heartbeat without touching the body, his or her body. And this is used as a measure of the sensitivity to perceive our internal body states and body signal. But what is more interesting is this now, this construct has been expanded and related to higher order social and cognitive dimension. It has been related even to metacognition, to executive functions. And in this last, let's say, last dimension, it is included also the modulation of attention to different body signals. This construct has been called with different names in the literature, but recently Schultz and colleagues defined this ability to focus the attention on a specific interoceptive signal for a specific time of interval as interoceptive attentiveness. And that's what we will be speaking about today. In our task, we manipulated this dimension. Interoceptive attentiveness was formerly conceived as a higher level dimension that could also be regulated and manipulated, but also trained through different type of practices. And between these type of practices, there are mindfulness-based intervention, controlled breathing, or even short relaxation treatments. It means that when we practice mindfulness-based intervention, we also we also modify our attention, of course, to this to the internal body state. That's obvious. I know that's super simple, but I think it's not still well known that at the basis of these attentional abilities, there is a higher complex and wider construct that is in general the awareness, the sensitivity, the accuracy, and the capacity to perceive the inner body signals. Skipping to the relation between interoceptive attentiveness and higher level cognitive functions, what has been demonstrated in the last years and what has been published in special issues recently in Trending Neuroscience of this January is that the consciously focused attention on breath, for instance, that stands behind these practices, can improve several cognitive and emotional processes. So even the simple focus on breath, it has been demonstrated to have an effect on sustained attention, cognitive monitoring, a metacognitive level of awareness. For example, these are just examples of cognitive processes that are related and referred and focus towards interoceptive inputs. It has an impact also on the regulation of emotions. So these practices can manipulate and regulate and reduce, in some cases, the negative affect. And what is even more interesting is that higher level of interoceptive sensitivity and receptivity have been even related to empathy. So there are differences in the empathy processing on the basis of in our interoceptive sensitivity and our interoceptive awareness as measured with the task that I've mentioned before. We recently tried to study even interoceptive attentiveness in relation to the observation of pain in others. So we manipulate the focus on ourselves while observing somebody else that is suffering. And in this sense, we wanted to observe how this ability can expand the empathic resonance of empathy for pain. And this is interesting also because a study with Meg demonstrated that the capacity to the development of interoception is related also to the development of empathy for pain. So we want to do the we want to study these processes in adults to see if higher focus on one's body and one's breath could expand and enhance this effect of empathy in healthy older in healthy adults. And in the end, what was seen was that the manipulation of HIHA was able also to manage stress response. In this case, as understood as the ability to regain concentration after a high impact stressful event. So this is a topic that was a bit ignored before, but now became always more important because it's combining the focus on the body with the higher level cognitive processes, as you can see, also with metacognition, that is another construct that is less studied from the neuroscience perspective. And since we are here, we are interested in speaking about NIRS and our NIRS paradigm. I've just reported some evidence from the article that I previously mentioned that regards the fact that previous neuroscientific research have also demonstrated that there are different portions of the right hemisphere, in this case, that are active while the person executes some interoceptive attention and awareness tasks. There are different hubs that respond differently for different aspects of the interoceptive processing. Just to give you some example, the supramarginal gyrus is considered as the neural coder of the body condition and of the peripersonal space, so it's more related to the somatosensory aspect and processes that going on during these tasks. But the interesting thing is it's the right portion of this structure that is active during these tasks. Also, the prefrontal cortex, the right prefrontal cortex, will seem to support more the aspect of sustained attention to the breath in this case, but in a goal-directed way. So the ability also to reorient the attention when is needed. And also the dorsolateral prefrontal cortex support these processes. In particular, there is also an involvement of the right frontopolar cortex to evaluate the course of action, the resource allocation to the situation, but also to this activation is also related to the exploration of the external exteroceptive inputs in this case. This is just to say that even if before the subcortical structure and the anterior cingulate cortex were believed to be more important to support these processes, now we know that even the prefrontal parts portions are involved, and in particular the right portions are involved in this process, in this high order capacity. What's even interesting for our study is that in particular the dorsolateral prefrontal cortex is also an area that is associated and that support social processes, including fully conscious motor actions, the capacity to adapt a different rhythm in a social condition, but also it was found active in cooperation among different partners, cooperative condition, as I said, interpersonal coordination. It has a role in all the social dynamic processes. Just to go quickly through the task in order not to leave you without this description. Today what we know is that there are a few studies, there are no studies that are focusing on how the manipulation of interoceptive attention impact on synchronization tasks. So this ability, this construct has been studied only in cognitive and simple tasks, but not in joint condition, not during communication processes, teamwork dynamic, or interpersonal relations. And what we want to see with our study that is now that now arrives to collect 16 couples, but we are still going on with the data collection, is to observe if the manipulation of this ability will impact even simple synchronization tasks. So we want to observe the effect of this manipulation on the dyads. For doing so, we apply EEG and biofeedback recording, but also a co-registration of FNIRS and EEG paradigm. For the NIRS montage, we are focusing mainly on the prefrontal cortex portions, and we are collecting the same two signals as mentioned so far. We are splitting the channel, as you can see in the pictures, the searchings and detectors on the two participants, and we are asking them to perform different tasks. In this case, it's just an example, but this is in our lab an example of a picture in which we can see different tools applied to the different participants. We use, of course, FNIRS CAP that are adaptable to EEG and NIRS co-registrations. In the experimental procedure, after a short baseline of 120 seconds, we start with different simple cognitive, linguistic, and motor tasks, of different type of task, but the duration and what is in common between these tasks is the instruction that the participant receives at the beginning of the task. In particular, we ask them to, in one case, just to execute the task in the control condition, or in the case of the explicit manipulation of the interoceptive attentiveness, we ask them to focus their attention on their bodily sensation, in particular on their breath, while they are executing a synchronized task. As I said, this task can be linguistic or motor. The difference between this set of tasks and this set of tasks is that the first regards just a linguistic cooperation or a joint motor act, while in the second and randomized block of tasks, the tasks are socially framed. In this sense, they are told that the way in which they will perform this task will be used to evaluate their capacity to work in team or to synchronize with a partner, and this task will also help them to increase their ability in this sense. I know I'm going quickly, but I want to show you that, of course, we are not only applying all these tools for the neural responses, but we are also asking them we are measuring their interoceptive sensitivity index with the heartbeat detection task at the beginning to check if there are differences between the couple in their ability to perceive their breath and their body. We are also applying several distinct measures to self-report measure to measure interoceptive awareness, because there are not so many questionnaires so far to measure explicitly interoceptive attention. We also apply a questionnaire for BIS, ERI, for emotional empathy and cognitive empathy, such as BIS, ERI, and also the questionnaire of cognitive and affective empathy, just because we saw and we knew from the literature that it seems that there is a relation between the ability to empathize and so maybe to synchronize and to tune with the other partner on the basis of different levels of interoceptive focus and interoceptive sensitivity. And what I wanted to show you today was that we just analyzed some data from a first pilot phase, in which at the beginning, before this this paradigm, we did some trial to check if this methodological implant could work. We, at the beginning, just asked to a single person to imitate an experimenter that was executing these simple linguistic and motor tasks. And then we started with the social manipulation and all the other complex steps. And what we saw from this result was that the explicit interoceptive attentiveness condition activates more the prefrontal cortex in general in these participants that were less from all the couples that we are now collecting. This manipulation also have an impact on the right hemisphere, but this right activation was only found in the linguistic task and not in the motor task. This is interesting because we know that the linguistic abilities are located even in the and are supported by the left hemisphere. But a possibility in this case is that the right hemisphere is here responding more for the type of manipulation that the participants were required to perform, to the interoceptive attentiveness manipulation. We are still going on with this study. We didn't finish with the data collection. In fact, the additional the next steps concern the increase of the sample size, and we still need to start analyzing the data for exploring the couples, the hyperscanning paradigm, and we want to explore the interbrain connectivity during the different conditions with a specific interest towards the socially framed tasks. And what we believe is that, and what we would like to test is if and how this focused breath on with this focused attention on our breath is really able to promote synchronization, not only in this basic, with another partner, not only in these basic tasks, but in general, even in more social processes. This is we can skip this part. Is only about the fact that these the FNIRS hyperscanning paradigm are useful for these social neuroscience approaches, but new and more studies, of course, are needed. I thank you for this attention. I know I'm just a little bit over after the time, but I'm here for just like some questions if there are, and I also leave our contacts, our email contact if you are interested in to collect, to know more about our study or to have some article or all the rest.
Very good. Very good. Thank you very much. I think we have a few questions from the audience. Anyone would like to start? Ishan, you're on mute. Unmute. Okay, while this is happening, I think I wanted to ask about, first when you asked about the cooperation and competition, the second study, yes? And do you think there will be or any effect measured by the FNIRS for reciprocating? Like they are not necessarily cooperating, but they are competing, and then when their behavior change toward being cooperating, do you think there will be an effect measured on the brain? Or I know that maybe it isn't studied, but
do you mean in the same task? Because these two conditions were divided, so they didn't switch.
Oh, okay. So it's between subjects. So each subject did something differently. Okay.
Yes.
Okay.
Yeah. So there are different couples doing different, participating for the cooperation or either for the competition study.
And second question I had, so for the study on mindfulness, this is fascinating. Is there anything surprised you so far from the results you studied? Like it seems all what you expected actually measured in the brain. Is there something surprising?
Well, I think that we would like also to check the behavioral level, because at the moment we are just observing that from a qualitative point of view, that the participants are referring that after the social framed task, they felt really more connected with the other partner. So what is surprising us is that really simple cognitive synchronization task could have this effect, as we know, on the empathic feeling. There is one thing that is surprising us, that is the fact that in the social frame task, in the linguistic condition, they feel more involved at the empathic level. They feel more connected between each other, but their behavioral performance and the cognitive performance are lower. So it looks like the task is more complex, too complex, because it's requiring to focus on the breath, to syllabize, and actually the social frame task in the linguistic condition concern the fact that they have to compose a word by syllabizing the syllab that compose the word, like synchronize, synchronization, etc. And this is what we saw, is that after this task, they feel highly connected, and they self-report that they are really, they love this task, and now they want to work with the partner. But at the behavioral and cognitive level, the performance is lower.
The opposite.
Perhaps because it's too difficult to focus on their breath, to be socially framed, and also to syllabize and compose a word. But the point is that sometimes when we do some tasks in our real life and in the daily routine, it happens that we are facing conditions like that. And we believe that if you focus even on your body when you're doing that, or if you're trained to do that, maybe at the end, not in a single task condition, you can be more able to really get connected with another and to perform better. So we should think about manipulating and changing something at this point, because we really believe in the idea, but the data that's surprising.
Sounds good. Ishan, is microphone working, or do you want me to ask the question?
Can you hear me?
Yeah, we can hear you. Yes. Go ahead.
Oh, great. Thank you, Laura, for a great presentation. I really enjoy your presentation from the very beginning. I know it's a lot of information that and a lot of questions, but because time is limited, I just want to ask one question. Because previous slides you explained about that there is so the mental effort when two people having competition is much higher than when these two people have different goal. Because I'm doing hyperscanning in the lab, but we focus by incorporating VR, and also we did in offline. I'm wondering, we know there's a lot of research has been done to see competition and non-competition, and they show different interbrain synchrony, or they have a different brain pattern. I'm wondering, in your research, why it could be in that situation the competition give more mental effort than when they having different goal. Does these two different conditions provide different brain pattern as well, or like a different brain mechanisms that may impact the interbrain synchrony? That's the question. That's interesting. I think your presentation for that.
Thanks for this question. Actually, I need to be honest, as has been at the beginning, this is not my study, so I don't want to tell you something that is wrong. But in this case, so the task concern similar goal that was to win, but so it's not completely divergent, it's not a divergent task. We do believe that the difference is really about the fact that they want to win or to demonstrate to be the best in this dynamic. We do believe that there is also an effect of the social feedback in this case that they receive during the task. So we also saw in the cooperative task that some interesting effects became significant after the social feedback, they received the social feedback. So we believe that this aspect had an impact in the interbrain synchrony and also in the behavioral performance, more than the fact that we're diverging or they receive a common goal, but they were at the end competing. So it was like as if there was an increasing effect during the task, and that this effect was mainly enhanced by this feedback.
I see. All right, thank you. Can I just ask one more question, Aladdin, in case we have time? I just want to add another question because Aladdin raised questions about mindfulness, which is, very like happening in the world of psychology. I'm wondering, because I missed a little bit in that part. Did you find any like interbrain synchrony? Maybe if you run an experiment when two people have like having like mindfulness related tasks, if in case if it does, then what do you think the factors that may lead? So for example, if two people are practicing mindfulness, maybe to calm themselves, what could be the most like leading factor that caused the interbrain synchrony among themselves?
Okay, thank you for this question. The fact is that this is the first task we performed, so we didn't perform this kind of hyperscanning approach on these type of participants. Instead, for our study, we are we are recruiting participants that don't have a previous meditation practice, because we are not so, we know that this perception stands as the basis also of brief relaxation practices and mindfulness, but we are not interested in not only in that, but more on the fact that even a simple focus on how we feel in the moment in which we are synchronizing with the other partner could have an impact on the interpersonal synchrony. So I hope I answered, but I don't have any, let's say, data or result to discuss because we didn't conduct this study. We were more interested in what stands at the basis of thinking about the body when we are doing something. Okay.
Right.
Thanks, because that's a very question. This is a very interesting question, but I think there could be some studies in the literature about that, but it was not specifically, let's say, our focus.
Sorry. Thank you, Laura.
Thanks.
One more question.
Yes. Hi, Laura. Thank you for a great presentation. It was really amazing. I really enjoyed it. So I have a bunch of questions, but just trying to summarize it and ask this. So I have read few papers of Professor Balkany, and most of the papers that I'm doing, my research is around emotional recognition in general. So most of the papers that in your lab release is about discrete emotions, so happiness, sadness, like this. So I didn't see that much, for example, dimensional emotions, like for example, talking about valence, arousal, or something like this. So first of all, is there most of the paper again, for example, as you said about positive emotions happens, for example, in left lobe or negative one, right lobe. And this is controversial because of different study protocols. So sometimes it's exactly become vice versa in different labs because of the studies. And but when it comes to discrete emotions, it's become a little bit easier to detect emotions because you just define whole pleasant emotions as happiness and unpleasant one as sadness or something, not happy, like this. So it's just binary classification. Is there any reason that because and all your studies, most of them is based on FNIRS. Is there any reason that you are using just these two factors, like pleasant and unpleasant, or there is no reason, just it's a procedure that you took?
Okay, thanks for this question. That's very a basic question in the sense that I believe that for my point of view, it's just a procedural step. We just consider these two clusters, but I do believe, and I don't want to reduce my answer to this, because I do believe that there is a reason, or there could be a reason that maybe also the professor will be able to explain. In my opinion, and what I've done so far, it was just to follow this step and to clusterize, to observe the general effects. So I do believe that there could be a distinction. I agree with you, and I know about the differences there could be, in particular in the right hemisphere, for negative and positive emotions. So I'm fully aware of that. But in general, what I can say without going in depth, because I don't want to say something wrong or too simple when it's not, is that I just I've proceeded like this because for me it was a first basic procedure to see if in general there are differences between these clusters.
And about the papers and studies that you run in the lab, most of the stimuli are short and they are not, for example, video-based. And the papers that I have read, maybe I'm wrong, just correct me if I'm wrong. Most of them, you don't have information about the stimuli that you use. So, and some of them is like, for example, using, if you, as I said, you are using like positive negative format, you are not using dimensional. So it's a little bit easier task in terms of detecting the emotions. So normally, for example, when you're running the study, you have, for example, if there is a study about negative and positive emotions, you are showing consecutively three negative pictures, and again stop and like positive, like this in the kind of blocks. And you don't have that much information. You didn't provide that much information about the stimuli in most of the time. I'm wondering about because there's a delay between EEG and FNIRS. So my project involved with FNIRS and EEG, both of them together. And as you know, because of the delay between EEG and FNIRS, I was wondering how we can, what's the, for example, the standard duration for stimuli? Because we say if there is 8-second or 12-second delay between EEG and FNIRS, we should consider that that person is starting to, build some emotions. So
Yeah.
emotions will not happen immediately unless you are using some harsh content. So since it's experimental room, experimental condition, and that person well aware of everything in the environment, it's a little bit hard to induce that subject to have those emotions. So, and consequently is expecting some, for example, vascular changing in brain
Yeah.
is a little bit far-fetched. So can
Yeah.
you please a little bit, walk me through this, what kind of stimuli duration standard that you're using?
Okay, thanks for this question because it's really interesting, and we are still working on that, even if we use FNIRS from since the 2015. We also use some video, actually. I don't know, like for
Short
the gestures, like for the gestures studies. I think there are also the videos are available in a journal of motor behavior, in the paper published there in Journal of Motor Behavior. And in general, we know exactly the same you said. We consider around 8 seconds for the stimula.
And normally,
For
because
the
you are using motor imagery part, so it's involved with the with participants' movement sometimes. So movement, it's it's a little bit visible in the brain when you are doing some small movement event when it comes to the FNIRS. But when we are talking about solid subject that's just watching the video, and we are expecting that subject showing some affection in the brain or something, a reaction in the brain, it's a little bit hard to realize if there is something. So that's why in my case, for example, I thought, since I'm not using any interactive movement or something, motion, so I thought, for example, 80 seconds would be, I mean, well enough to just capture FNIRS and EEG if it happens at all. So
okay. So you mean in a video, 80,
yeah,
you said 80
80,
seconds.
yeah.
In
this
I
case,
think, well, it always depends on what you are studying. It could be enough, but have you already tried to with a pilot and to see
yes.
if you fall?
So
Okay,
so
and 80 was.
So you think it's small or it's long?
I think it's long. I think it's long.
Mhm.
We use like 1 minute for some videos, but as you said, even less. Well, it depends. It depends on the study. Like in some studies with advertising, we use 1 minute.
Deep data set, as you know, it's just 1 minute, 60 seconds. So the reason that I use 80 seconds, because deep data set is not good enough. It doesn't show that much effect. So we have to use a lot of machine learning or deep learning models to understand the emotions or some patterns out of the data set. So even in 80 seconds, I kind of manage the video somehow that it's fading and fading out like this, to make that person starting to feel something, build something. So it's not like deep data set. Deep data set has just some video clips, like music clips, and it was just suddenly start and suddenly turn down the things, whole video. So it doesn't show anything. But in my videos, I'm just starting slowly and finish it slowly to allow that person to settle down and induce himself in terms of emotion.
And then
Exactly.
So, understood what you mean.
Yeah. And last question. Sorry, Aladdin. I think
I just want to check. You have to go somewhere else. Yes, you have. Laura?
Yes, I have a class right now, but
Sorry.
I think we
can send you the messages by email, the questions. We have more questions as well. Okay.
Yeah, of course.
Thank you very much.
Thanks, you too. It's really a pleasure. And of course, we can also talk about that. I don't want to say the wrong name, Alireza.
Yes.
Okay,
yes.
Thanks.
Thank you.
Thanks for your question, and thanks
really, it was amazing.
for your time. And also to Kunal that wrote in the chat, I can answer by email, but yeah, I have another class, that's why I need to leave.
Sounds good. All right. Thank you very much.
Thank you.
Thank you so much, and bye. Have a good day.
Yeah. Ciao, ciao.
Thank you.